Thanks for inviting me. I really appreciate it. It's my first time in the Southern Hemisphere, if you can believe that. So I'm very excited to be here with you all and talk to you about this. I'm going to introduce you to some work that I started a few years ago now as a postdoc, and I've continued in my new lab at Idaho State University. And let me just preface this by saying that a global pandemic is not the ideal time to start up a genomics lab. And so it's taken us quite a bit, but we now have some preliminary results. And so everything I'm going to show you today is either or both preliminary or on a small data set. So, don't hold me to it. Things might change in the future. Okay, so I'm going to describe several ways that we are trying to connect genomic data with other types of data throughout our work on single invasive weed species. So first, let's get into the weeds. It's an invasive plant joke. So this is a photo of an agricultural field in Wyoming in the US, and it's infested with a species called blue mustard, who's the star of our story. So everything that's purple in this photo is blue mustard. Now, why it's not called purple mustard, I don't know. Good question. And when I see a landscape like this, that's dominated by a single species that we not only didn't intend to put there, but actually don't want there at all, I really want to understand what's going on. And so that leads me to my motivating question, which is, how do invasive plants succeed in novel environments? So I want to understand how populations introduced to novel environments outside of their native range can survive and thrive, and what happens to them once they get there. As has been mentioned, invasions are these really great evolutionary test cases. And I really think of invasion biology as the biology of winners, even though we don't like that they're winning. We're Sherlock Holmes and they're Moriarty, right? So with invasions, we can ask questions about the role of standing genetic variation, admixture, or novel mutations in the process of adapting to novel environments. And some of the questions we typically want to understand are invasive species sort of invasive before they're introduced? In other words, are they pre-adapted or ex-apted to similar environments? And that's what allows them to hit the ground running. Or do introduced species evolve to be more invasive after introduction? And then how many introductions, or what's the propagal pressure that's really required for an invasion to stick? And one way we try to answer these questions for a particular species is by collecting individuals from the native range, collecting individuals from the invasive range, and comparing them and saying that any differences we see are the result of evolution in the invaded range. And I've done work like this myself. But I want to ask, can we actually make better comparisons? Because there are challenges to comparing contemporary individuals from the two ranges and then saying that the difference is evolution in the invaded range. And many speakers have mentioned some of these problems previously. So I'll just add or reiterate that it's often not clear which population from the native range we should be using in our comparison. And this can be really complicated if there are multiple introductions, which seems to happen quite a lot, or if we lack detailed population genomic data, which, as Paige mentioned, is a big problem. And additionally, it's not like evolution has just stopped in the native range. Global change is global change, it's happening everywhere. So ideally, we could compare ideally, we could observe the direct predecessors of the contemporary invasive populations that we're interested in. And so what we need to do that is just a time machine, right? No problem. But in a way, that's what we have in the form of herbarium specimens. So maybe I'm being a bit dramatic about herbarium specimens, but I want to take a moment to really make the case for natural history collections-based research, just in case you aren't already completely sold. So natural history collections, of which herbaria are some of the largest and most accessible, have provided the data that is the foundation to our understanding of changes in biodiversity over the last couple hundred years. And herbaria are these really rich sources of primary data collected by the original collector, such as label information. This primary data can be super useful, and people have used it to do some really interesting work over the last several years. And what I think is really exciting is that even more data can be collected from these specimens. So this novel data It can be collected from digital representations of the specimens or from the physical specimens themselves. And one of the many kinds of data that I can get from an herbarium specimen is genomic data. Across multiple specimens, all of this genomic data is temporally ordered. And using the temporal nature of this genomic data, we can start to understand key population events in the history of a plant invasion. So with time-separated genomic data, we can really directly observe or watch genetic variation as it changes through time and as a species spreads across environments. For just a handful of invasive species, we have an idea of the levels of genetic diversity that currently exist in the contemporary invasive populations. So we can see this sort of like endpoint. But by using the historic record, we can start to understand some hidden patterns. Something we'd really like to know is what triggers an invasion. Does an invasive species start to spread just as soon as it arrives in a new habitat? Did just like a single or a few populations show up in the new range, and that's all it took for it to spread immediately? That's sort of the simplest case, like a continuous population model. Or, as is often thought to be the case, is there a lag period? And what's a lag period exactly? Before the invasion can really take off? And what causes that lag to end? So perhaps there are multiple populations that are introduced, and then a sorting occurs, so that only the best suited genotypes remain in the invaded range, and they replace all the others. It could be this replacement event that triggers the end of the lag period. Or possibly populations admix, and that increases the genetic variation available. It would then be this admixture event that ends the lag phase. And of course, some combination of these things can happen as well across the invaded range. For a lot of invasive species, there's just too many unknowns to really get at these types of questions. And I think that's where herbarium genomics can be really useful. So my focal invader, I'm starting with Chorispora tenella, or blue mustard. It's also known as musk mustard because it's very smelly when it flowers, sort of the unwashed scent of humanity, I would say. You can smell it before you see it. And there's a few things about this species that make it the perfect focus for this kind of work. First of all, it's weedy. It's a weed of winter annual cereal crops like winter wheat and alfalfa. When dairy animals graze blue mustard, they produce milk that is bitter in taste and foul in odor. And it's also a weed of arid rangelands and roadsides. And it's one of the 3,000 species listed as a weed by the Weed Science Society of America. Most of the work that's been done on the species in the past has really been focused on exactly what herbicides kill it and how quickly. So the species is native to Eastern Europe, but also the Balkan Peninsula, Turkey, Iran, Mongolia, northwestern China, and northern India. And now it's in North America. So it's been observed in 31 of the US states and in 3 Canadian provinces, including everywhere west of the Mississippi River. Now, I did do a quick little search to see if it's in New Zealand, and you guys are in luck, I don't think it is, based on my little search. Good job. But it has been observed in Australia, so maybe watch out for that. Okay, so blue mustard is well represented in North American herbaria. It was introduced at a time when it could have been collected by botanists throughout its invasion, so it first showed up in the early 1900s. And indeed, they did collect it. So this is the cumulative curve of herbarium specimens collected in North America. And these are just the ones that are listed in the single large database. So this is going to be an underestimate of true herbarium holdings. And finally, the species has some really convenient characteristics from a genomics perspective. So there are significant genomic resources available for closely related species because it's very handily related to everyone's favorite model plant, Arabidopsis thaliana. It's also a diploid, and it has a relatively small genome for a plant. So it's about 680 megabases, which is twice the size of Arabidopsis. And believe me, it could be so much worse than that. So this is almost the ideal situation. The species is also an annual, which means that there has been more opportunity for recombination since introduction relative to a species that would be a perennial. And this might give us the ability to identify causal genes underlying selective sweeps. And given all of these characteristics, I think this work could be especially informative because we should be able to drill down to the functional level to really understand how the species has evolved since introduction. Okay, so to help my genomic analyses along and to make sure I even knew what I was looking at in the sequence data, I needed a reference genome, even a really bad reference genome, compared to DNA extracted from fresh tissue. Historical or ancient DNA is typically weaker, it's more fragmented, and has some characteristic damage patterns. And without a reference genome to compare to, when I looked at the sequencing data from herbarium specimens, I wouldn't necessarily know if I was looking at Chorispora, if I was looking at some other contaminant species. So I put together this draft reference genome assembly using a contemporary individual. And this is a very rough draft that still needs work for sure. So I got the DNA for this assembly from an individual I grew up from a USDA germplasm population. So this was originally collected in Iran, so it's part of the native range. And, you know, like I said, it needs some work still, but it's useful in some contexts. So once I had this draft assembly in hand, I could proceed with working on the herbarium specimens. And I wanted to see how many herbarium specimens I could actually get my hands on, how many they would let me sample. And it turns out it was kind of a lot. In my experience, herbarium curators are not too bothered to let you rip leaves off of sheets of very common invasive plants. So your mileage may vary, but I was pretty successful in this. With the help of just 9 herbaria, I was able to sample leaf tissue from about 750 specimens that were widely distributed across the Western US and parts of Canada. But there's definitely more out there, and I would love to sample them as well. And each of these specimens represents not only a point on a geographic map, but also a point in time and in environmental space. And I think the ultimate goal of this work is to understand the invasion process across these several dimensions. Okay, so you can see sample density in my lovely GIF that I made here. And it's binned roughly by decades, so the decade's up in the corner there. So the earliest specimen that I have in this data set dates back to 1916 from Boulder, Colorado. So that's in the middle of the country there in the Rockies. Within a few years on either side of this specimen, there's also a collection from the West Coast in Washington and from the East Coast in New York. And that suggests to me that there may have been a few separate introductions, but I would want to look at genomic data to be sure. Before I get into the genomics, I just wanted to show you a couple of my favorite specimens. So this is the earliest specimen that I've found so far. It's from New York in 1894. It was collected from about the Yonkers factory grounds. And my understanding is that there was a wool sorting factory in this location, and that maybe that could represent the first invasion route into North America. And then there's this one, which always makes me laugh. This was collected by a botanist out of Pocatello, Idaho, where I'm located now. And I'm told his name was in fact pronounced J.H. Christ. But you know, it was a choice that he named his herbarium that. And I particularly like that this species happens to be a mustard, which used to be called crucifery because of the cross-like shape of its flowers. And in fact, this species, one of its many common names is crossflower. So, you know, this guy really nailed it. Okay, back to the genomics. With the help of some collaborators of mine at the Max Planck Institute in Tübingen, I got to pull on this, you know, extremely fashionable outfit. And we produced sequencing libraries for a pilot set of 20 herbarium specimens, ranging in time from 1941 to 1993. And these were collected from Nevada, California, and Colorado. And we then sequenced these to low coverage, getting about 10% of the genome back to look for patterns of ancient DNA damage and non-target species contamination. And this is what we found. So the y-axis here is sequencing, number of sequencing reads, and the x-axis is each bar represents an herbarium specimen, and there's 2 library production blanks included at the end there. The dark brown color indicates that the majority of reads map back to my modern Chorispora assembly, so contamination relatively low. I also checked for common ancient DNA damage patterns, like C to T conversion, and we found relatively little damage. So since the low coverage libraries all looked pretty good, we sent them off for much higher coverage sequencing, about 33x, and that's the data that we've been working with so far. And very excitingly, we have some super fresh preliminary results from this pilot data set to share. So this is the result of some analyses done by Therese Balkenbush, my PhD student. And this is an unrooted consensus tree using the NeighborNet method in SplitsTree. Based on their chloroplast sequences, this is how these 20 individuals in the pilot data set are related, and I've colored each of the samples by year. So this is a very small data set. We're not going to answer our questions with just this, but there are some intriguing bits here, I think. So for one, there is some chloroplast haplotype diversity across this data set. So we know there wasn't just like a clone that was introduced to the invaded range. Also, remember the strength of the temporal sampling. For example, this little cluster here includes individuals that range in time from 1940 from the 1940s to the 1980s. And they're all very closely related or have the same chloroplast haplotype. And so that tells us that at least this one haplotype has remained in the invaded range during this time. So it hasn't been replaced, at least over that window. And it was present in all 3 states that were in that data set. So definitely, I want to expand this data set, and we are working to do that now with the help of some collaborators at Penn State. So they're hard at work on this now, and this expanded data set will include some of our earliest specimens and focus on locations that are well represented in herbaria, so counties that have many specimens collected in them over this time period. And ultimately, what we want to do with this whole genome data from these specimens is understand how genetic variation in the species invaded range has changed over the course of the invasion. Okay, but plant genomic data isn't the only kind of data that you can get from an herbarium specimen, because herbarium specimens are awesome. They're these beautiful little nuggets of data, and the more I squeeze, the more data falls out of them. So I tried to come up with a few additional ways that they could help me address my main question. And I want to talk about how I'm expanding this project and what a rich data source herbaria and natural history collections in general can be. Okay, so to return to my motivating question, I want to know how invasive plants succeed in novel environments. And so far I've told you how herbarium specimens have allowed me to investigate how plant genotypes have changed over time, over the course of an invasion. And I couldn't do this without collections because the time series sampling available from herbaria is unparalleled. But of course, there's lots of information additionally available on the label of a specimen. This is an example. And particularly useful is the information about the individual's abiotic environment. And we can use just this information to address questions about how species are distributed across the landscape and through abiotic environments. So this approach is particularly powerful when you combine it with large environmental data sets. For example, using occurrence data taken from herbarium specimen labels and combining that with environmental data taken from satellite images, my collaborators and I were able to look at where species in a particular genus occurred on the landscape and associate that with drought frequencies in those locations. So in the study, we found that annual species in the mustard genus Heliophila more often occur in areas that are prone to drought at a time of year when the plant is in its seed phase, so it's more drought resistant. And that perennial plants in that same genus tend to occur in places that are less prone to drought. Now, this particular study was in a genus native to the study area, but I think it illustrates how useful herbarium specimen label data can be to understand the abiotic environment that a species encounters. So I'm also trying to connect historic patterns with genetic variation we observe in contemporary populations, right? And I can use the label data from herbarium specimens to focus my collecting efforts. So the counties highlighted in blue here blue and green. These are the counties that we're focusing on for the expanded plant genomic data set because they're the most well represented in the herbarium specimen collections that we have. And I've also recollected fresh material from populations in these same regions. And the black triangles represent contemporary collections from those places. So having contemporary collections from these places will help me draw conclusions about how things have changed over time. Contemporary populations are amenable to certain things that herbarium specimens just aren't that great at, like you can sample way more individuals in a contemporary population than you can typically get off an herbarium sheet. And so that's really helpful for population genetic analyses. And also I can get seeds from contemporary populations. And once I have seeds, then I can do elaborate common garden experiments to try to associate phenotypic variation with genetic variation. We actually have some sequencing data back from some of these contemporary populations. And I got to work with an undergraduate researcher over this past summer, Rachel Havoc, to see how these contemporary populations are related to each other. Now, she ran a principal components analysis of the genomic data for these contemporary populations to see basically how they're related. And this was just a practice run. So this is just a subset of the data that she was working with. So this figure will definitely change in the future. But she's rejoining the lab in the spring so we can keep working on this project, and I'm really excited to see what she finds. Oh, sorry. Okay, so in addition to looking at genetic change during invasion, I can also use herbarium specimens to look at how the geography and abiotic environments that the invader encounters also change during invasion. But with herbarium specimen sheets, we also have the individual right there. So we can get data about phenotypes. And that's the level that natural selection acts on directly. And so that makes my little ecological, evolutionary ecologist heart, go pitter patter. Okay, so I've been working with Mason Heberling at the Carnegie Museum of Natural History to see what we can learn from the phenotypic data preserved in the herbarium record of this species. We're interested in changes in phenotype that occur over the course of the invasion or in association with novel habitats. So here I've shown you some of the raw data of a phenotypic measure, in this case, day of early fruit, which we've collected from images of herbarium specimens to understand what time of year Chorispora starts to produce fruit. Now, the best thing about this type of data is that it's completely non-destructive, like no ripping of leaves off required, and it's remotely accessible. So if an herbarium has scanned an image of a specimen, we can use it. And one of the things you can do with this type of phenotypic data in a temporal context is ask questions about the abiotic environment that produce these plant phenotypes. So we're asking, how does regional climate influence phenology across the invaded range in the species? The data set for this project included 528 images of herbarium specimens from multiple herbaria, specimens collected across Western US between 1920 and 2020. PhD student Rachel Reeb at the University of Pittsburgh has collaborated with us to do some really cool analysis with this data set, asking what environmental factors determine when a plant will reach a certain phenological stage. And what she found was that mean annual temperature and interannual temperature variability were the most predictive of phenology day of year. So in other words, what day of the year is it when a plant reaches a certain phenological stage like flowering? So the average timing of phenology across all life stages occurs earlier in warmer regions than in cold regions. So that's not really a huge surprise. But this interaction with the interannual temperature variability is kind of intriguing. Okay, so this is a plot of the model outputs. And for plants that are present in the coldest part of the range, sorry, let me back up. So this is mean annual temperature gradient on the bottom and phenology day of year on the y-axis. And the different colors represent different amounts of interannual temperature variability. Okay, so plants in the coldest and the most temperature variable regions experience the most delayed phenological timing, likely serving as a strategy to avoid winter frost. And that translates onto this map that the plants in the locations with the yellowest dots get to a given phenological stage about 100 days earlier than plants in the locations with the reddest dots. And considering that this plant only lives like 4 or 5 months, that's pretty impressive lag across its range. So interestingly, the interannual temperature variability levels were less diagnostic for the warmer parts of the range. Let me get this part right. Okay. So the warmer regions have long growing seasons, such that temperature unpredictability might not impose such a large risk based on frost for the plant, and thus have a reduced effect on phenology. So phenological variation along this mean annual temperature gradient is contingent on the predictability of the temperature regime itself. And as far as we know, this kind of pattern hasn't been shown before. And it may have some important implications for this plant under climate change, which is supposed to both increase temperatures and unpredictability. We think that this spatial pattern is driven, at least in part, by some adaptive genetic variation in the phenological strategies of blue mustard. And potentially we can identify the underlying genetic variation here because we are sampling genomic data from many of these specimens. Okay, so we can understand how genotype and environment interact to produce phenotype, and that's cool. But wait, there's more. Because just the abiotic environment doesn't perfectly describe the novel habitat that an invader is going to encounter. There's also a whole complex system of novel biotic interactions that an invader has to contend with, and that may enable or resist invasion. And I think that herbarium specimens can help us understand this as well. So one exciting possibility is that we can use herbarium specimens to understand the historical microbiome associated with invasion. By sequencing microbes from these specimens, we may be able to observe microbial communities that co-invade, that act as facilitators, or even suggest future biocontrol candidates for the species. As part of the Sea Grant through Penn State, we're using that expanded plant genomic data set to also investigate the metagenomics of those individuals. And so that's part of the ongoing lab work. Using genomic data from herbarium specimens, we're going to explore the historical microbial community using a pipeline developed by Laura Wyrick, which identifies bacteria, archaea, viruses, and microbial eukaryotes from ancient genomic data sets using a database of over 130,000 full microbial reference genomes. Because time damages all things, including DNA, we can use the characteristic damage patterns to separate modern contaminant microorganisms from historical microbial reads, so things that should be living on the plant when the plant was alive. And we're also using environmental swabs to assess possible modern contamination from storage. And in this way, we can start to understand at least a slice of the biotic environment that the invader encountered during invasion. We've started our metagenomic investigations by looking at that 20 specimen pilot data set. And so these are the 20 herbarium specimens that I wanted to get plant genomic data from. And so I wasn't doing anything special to collect microbial DNA, but nevertheless, in that data set, along with the plant DNA, that came all of this microbial DNA of things that were living in and on the plant or on the specimen. So at the sequencing depth that we targeted for the plant, we can be pretty confident that we're getting the vast majority of the microbial diversity as well. That's what this rarefaction curve is telling us. Once we get past about 100,000 reads, our diversity estimates aren't changing that much. And this is what the microbial diversity looked like for these specimens. So each column here represents microbial reads for a specimen, and the colors represent the proportion of that community in each of the top 10 represented bacterial clades. So these specimens are grouped by the herbarium that they're maintained in, and then they're ordered by year in this figure. So we were actually able to identify a very small handful of taxa down to the lowest taxonomic level. And these kind of reassuringly included several taxa known to live on the leaves of plants, which is where the samples were taken. So like leaf-specific Pedobacter and a plant pathogen, Pseudomonas viridiflava. So yay, got some stuff that should be there. And again, this is a very small data set, so we can't say, that much. But there does seem to be maybe, possibly, a trend towards increasing microbial community diversity with later phenology. So the more mature a plant is, the more diverse its microbiome is. And this is using that same phenology index that we use for the to look at the environmental effects of phenology day of year across the Western US. And hopefully once we have the expanded data set, so some of these patterns may become clearer. Okay, so to wrap it up, to answer my question, how do invasive plants succeed in novel environments? I think I can get a lot of the answer from the many types of data available via herbarium specimens, all of it enabled by collections and curators that have maintained these really impressive resources. Natural history collections like herbaria allow us to directly observe the evolutionary and ecological changes that occur over the range of an invasion, but also over time spans that are longer than a graduate degree or even a human lifespan. And so I think bringing the different streams of data associated with herbarium specimens together is really a powerful and exciting approach to understanding invasion and rapid adaptation to novel environments. And other folks are also starting to use this approach or similar approaches very successfully, and including people in this room. So I think it's going to be really exciting to see how it works out. And with that, I'd like to thank folks in my lab and my collaborators, the many herbaria that I've worked with, and my funders. And I'd also like to acknowledge the Shoshone and Bannock peoples on whose traditional land I now do my work, and to thank you guys for listening.
Thank you so much, Kathryn. That was really interesting. Does anybody have any questions?
Lovely talk, thank you. And this is a very question from a non-plant person, but is there any chance of seeds in the herbarium specimens that would still be viable?
Oh, yes, possibly. I haven't tested any of my specimens, but every time I see some seeds, I grab them. But I think other people have occasionally been able to germinate seeds of other species from some specimens. Now, the older they are, the less likely, right? But yeah, wouldn't that be really cool? Do like a resurrection common garden with like 100-year-old seeds? The dream.
Anybody else?
Thank you. That's super fascinating. I love squeezing out all the data you possibly can from those samples. I was curious, the microbial angle is really interesting. Is there also something you can get potentially from about the maybe parasite community or other things by like damage to the leaves or anything like that? Is there maybe even more you can squeeze out about the biotic selective community?
Yeah, I think so. So other people in other species have looked at herbivore damage, to be sure, on by measuring the bite marks on plants, and they can tell within some range of uncertainty what was the thing that was eating it. I haven't tried that with this, but I think that is potentially a way we could go. Yeah, that'd be really cool. Very cool. I don't know about parasites. What?
I guess maybe I mean I think you call them herbivores.
Oh, sure. Yeah, herbivores. There's also on some specimens, there's actually dirt on the soils. And so maybe we could get at, soil microbes as well. Yeah.
Did you have a question? Yeah. Okay. Anybody else?
Just to follow up on the microbiome, would you think about comparing them to anything contemporary, or do you reckon that's not going to give you something too helpful?
Oh no, I think that's the point, right? Because I want to know what was their starting conditions, and then how have they ended up now? because just looking at how they're doing really well now doesn't necessarily tell us about any of the challenges they had to pass. And so one of those might be recruiting new microbial facilitators, or increasing tolerance to new pathogens or something. And so I think the difference between the contemporary and the historical is really interesting and important.
How do you think you'll distinguish the, I guess, the decline in community over that time? Because microbial decline obviously is going to be something to manage, but maybe you have some species that cling on for a long time that you can use as reporters or something.
Do you mean like detection from the herbarium
Detection.
specimens?
Exactly.
Well, so the people I was working with in Tübingen, they were like a little bit bemused by my presence, because the stuff that they work on is Neanderthals and mammoths. And so they're like, why are you bringing this stuff in here from the 40s? Like, very unimpressed. And so, and DNA is really robust, all right? So I'm not super concerned that there's going to be a huge drop. I mean, we can maybe do some kind of study looking at that, but I would expect that most stuff, especially if it was in high abundances, would persist for 100 years. Yeah, that would be my guess.
Good luck.